Student, today in this video I'm going to start a new chapter called Heredity, okay, which is common both in class 10 and 12 standard. So the chapter name is Heredity, or we can also call it as the genetics. Now when we say genetics, genetics is a branch of science that deals with the study of heredity and variation. So it's one of a branch of science that deals with the study of heredity and variation. Basically, we are going to study about the heredity, but not the variations. Okay. So, when we say heredity, the character that has transferred from the parents to the offspring, or the character that transfer from one generation to another generation. Okay, that is considered as the heredity. Now, when we come to the term heredity, this is a very common scientist, well-known scientist, okay, which we call, he is known as the father of the genetics. In the modern science, the classical biology and the modern biology, which we call molecular biology, okay. So the genetics, the idea of genetics the credit goes to Gregor Johann Mandel, and that is why we call him as the father of genetics. He was born in 1882, okay, and then he worked for 6 years, okay. He had done research for 6 years on a pea plant, Pisum sativum. And then he died in 1884, okay? But his works were not recognized, okay? His works were not given credited. Even though he worked for 6 years on pea plant for Pisum sativum, his work was not recognized. Then later, 3 scientists, they were known as Hugo Vries, Correns, and Chermak. Okay, after 16 years after his death, he died in 1884. After 16 years, these 3 scientists in 1900, they gave credit to Mendel's work. They recognized Mendel's work. Okay, and after that only, we consider the father of genetics as the Mandel. And till that, today we are using his principle in the branches of genetics. Now, let's discuss why his work were not recognized. Okay, during that time, there was basically there was no journals. Okay, he did not publish his work in a larger scale. Okay, like today, if we do research, we update in a journals, and then all over the world, different scientists, different researchers, they study and they know about the other works. But during that time, he did not publish his work in a larger scale. So that was one of the reasons for his non-recognition. Okay. The second reason was he was a mathematician. Okay. Now, as a mathematician, he was explaining biology. He was giving the principle. Okay, explaining the inheritance of the character. Okay. Now, Mandel, he said that he termed that character, the inheritance of character, something is present, that is from one generation to another generation, something that passed, and then he named as factor. And that factor he considered as discrete. Discrete factors. So many biologists were not able to understand the meaning of it. Okay, and as I said, he was a mathematician. As a mathematician, many biologists were not able to understand his explanation. And the third reason was, during that time, the microscope was not that common. Okay, people did not know about the structure of DNA, how the inside the cell. Okay, they were not they were not able to study through physical observation about inside the cell that there is these factors are considered as a gene. So today we consider that as, we name this as gene, that factor is considered as gene as of now. And then this term gene was given by Johansen. But Mandel said that as the character that transfer from one generation to another generation, he named it as factor. So today we know it as a gene. Okay. Now that was the reason. Okay. And then during that time there was a very common research was going on that was on the basis of evolution, where the Darwin evolutions. Okay. Many scientists or researchers were talking and giving focused on the evolution, that is Darwin theory. And that was some of the reason for his non-recognition of his work. But later, after 16 years, because of that, those three scientists, some of the reason for the success of his works were, as I said, he selected pea plant, right? So that pea plant, in scientific name, we call Pisum sativum, and he had taken selection of contrasting character. Contrasting character means like he had taken 7 different threads of pea plant, which have different contrasting characters. For example, one pea plant has its seed color is just say round, color green. Okay, then yellow, for example. Okay, shape like round, wrinkle. Okay. Height like tall, one is dwarf. Just say the flower position, axillary, terminal, okay. The flower of a color like purple, okay, and then the opposite color was white. So 7 different contrasting traits were selected, and that is one of the reasons for his success. And second was large sample size. We all know that pea plant, its life cycle is only 3 to 4 weeks. Pea plant life cycle is 3 to 4 weeks. So in 3 to 4 weeks, we can produce many generation plants. Okay, so its life cycle is being too short. We can in just few period of time we can study many generations. And in that way, we can produce large sample size. The third was data analysis. When he was collecting the data, it was very systematic, okay, and it was in a form of paper presentations, okay, and that is the third reason, okay, for his success. The second question comes: why Mandel had selected pea plant for his experiment? Why not any other plant? And the first reason, the same point can be done is, it was the pea plant has a short lifespan. Okay, its life cycle is only 3 to 4 weeks, so we can produce large sample, or in a short period of time we can study many generations. The second reason is it can undergo self and cross pollination. Pea plant can undergo both self and cross pollination. You know what is pollination, right? The transfer of pollen grain from the anther to the stigma. Okay, so the pea plant can undergo both self and cross pollination. And the third was constructing character. Constructing character. as I say, he had select 7 different contrasting character, okay, like height. So one will be tall and another one will be dwarf, for example, okay, like flower position. One will be axial, another one will be terminal, okay, like seed color, okay, the one will be purple, okay, and the another will be green, okay. So those are the 3 reasons, okay, why Mandel had selected pea plant for his experiment. And the 4th reason was that the seed of the pea plant does not show any dormancy. The seed of the pea plant do not show any dormancy. And more number of seeds, okay? Number of seeds is more, okay? Number of seeds is large or more. Okay, so this is very important, okay? Why Mandel had selected pea plant for his experiment. To understand the Mandel law of inheritance, okay, we have to understand certain few terms, okay, that I'm going to discuss right now. The first term was gene. When we say gene, now in today's term in molecular biology or the present molecular biology, that is our genetics, the gene, as I said, it was known as a factor or discrete given by the Mandel. The term gene was given by Johansen, and it is also known as the unit of inheritance. The factor that transfer from one generation to another generation, that shows the character from the parents to the offspring. It's all because of gene. Okay. Second point that you have to know is allele. Now, allele is a form of gene. Forms of gene. Okay, and in allele we represent in the form of letter. For example, capital T. So this capital T, I would say this as tall character. If a plant has tall character, for example, a person may have tall character, okay? A father may have tall character, but a child may have short character, right? Dwarf character, tall, then small t as dwarf. Same letters, right? The allele for tall character, it is capital T, and for allele for small letter t, it represents dwarf character, okay, or short. So when I say allele, capital T or small t, it represents allele, okay. It means that there are 2 genes, right? 2 alleles, okay. Under that we have dominant. When we say dominant, it represents about the character. So dominant character and recessive character. So there are 2 types of allele, dominant allele, recessive allele, okay. So dominant homozygous and dominant recessive homozygous. So when I say homozygous, dominant homozygous or recessive homozygous, it means that homo means same, allele will be same, but dominant character. Now, in these present generations, okay, we prefer tall character than the short character, right? So that is considered as the dominant character, okay? So if I write capital T, capital T represents dominant character, But here, both the allele have dominant, both the allele is showing capital letters, capital T, then we can represent as dominant homozygous. Likewise, we can also represent capital T and small t, okay, recessive homozygous, okay. Next is heterozygous. So heterozygous means it is represented by capital T, small t, okay, but the character is dominant. We'll discuss why is it dominant. Even though you have an allele, if a person have an allele capital T, small t, the character will show dominant, okay. That will understand from the Mandel law of inheritance, okay. The next, fifth term that you have to know is cross. The meaning of cross. When we say cross, it's about breeding, okay. Male or female, okay, they go for fertilization and then a breed has been developed, right? So when we say crossing, cross, it's about breeding, okay. So just say male and then female, okay, for the fertilization, okay. After crossing, we have a gametes formation, right? So the sixth term will be filial. So sometimes we write F1 means F1 generations, okay. Just say I have taken a tall plant and a short plant. Just say capital T, small t, small t. So it will form a gamete capital T. So this will form a gamete capital T, small t. Okay. So this will consider as the gamete formation. After gamete, if we get the first generation, okay, after crossing, male and the female gametes, after crossing, the first generation that if we get it as a product, then we consider that term as filial or F1. And further, if that generations go for crossing, then we consider as F2, F3, and so on. Okay. So F1 means first generation or filial, first filial generations. So this is about the filial. The next is about Punnett square. So when we say Punnett square, it's about probability. Okay, the probability of the character. Okay, the next generation. For example, if I select one tall plant and one short plant, okay, what would be the probability of a plant that will produce, for example, tall plant? Okay, or what will be the probability that there will be a short plant? Okay, so Punnett square will help the probability of finding the character of a plant. The next is about phenotype and genotype. When we say phenotype, physical appearance, okay, physical appearance, like short, tall, okay, physical appearance. And when we say genotype, we talk about genetic makeup. So these are some of the important terms related to genetics. In the next class, we'll discuss about Mandel law of inheritance. Thank you.